it's a study guide sheet that you have in Blackboard for your third exam, just to make sure that you're studying all the right things and that you're not missing anything or spending too much time on something that it may not be all that important. So I'd say the first place to start really is with the DNA and genes. So again, conceptually, there's some specific details you need and a lot of big picture stuff. So the big picture stuff is, bacterial genes, right, that code for the information that make up the cell, are encoded in the bacterial chromosome. All right, and that chromosome is circular. Okay, so it's broken up into regions with many different genes, and I just put a couple different colors here. There'd be far more genes than this. Now, if you took apart, a piece of this, you would find that that is, double-stranded, we call it DS, double-stranded DNA. So if you really need to know what DNA is, starting back at the most basic level of the nucleotide. So a nucleotide is a 5-carbon sugar. There's 1, 2, 3, 4, 5 carbons. The 5th carbon has a phosphate functional group attached to it. The 3rd carbon has an OH hydroxyl group. In DNA, that's D for deoxy, so the number 2 carbon is missing its hydroxyl group. The number 1 carbon then has a nitrogenous base. And that base would be R, A, T, G, or C. And that's what makes our genetic code. The sugar is always the same, the phosphate's always the same. What's unique is that there are these 4 different nucleotides and arranged in different orders that then code for information. A strand, so a single strand, of DNA. So a single strand of DNA is made up of chain of the nucleotides. Okay, so we have a phosphate from 1 attaching to, let's just say this is A. The OH group of another. Now, this needs energy, so again, I went through this in all these lectures. This is typically a triphosphate, then the 2 are cut off, and then a phosphodiester bond is made, and so on. And then we make these chains of nucleotides. So a single strand is a chain of nucleotides held together with phosphodiester bonds. And then what we have are this particular chain. So if we reduce this down to, like, say, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, like this. So the phosphate's there, the sugar's here, like this. Then sticking off the sugars would be, the A, T, G, C, for example, OH group here. That's what you would have. Now, DNA, though, is double-stranded, right? Not single-stranded, so you have to know how is the other strand attached. So the other strand of DNA is going to be running like this. So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, right, like this. And so what we're going to end up with is an OH group sticking off here, a phosphate sticking off there. And if we're going to look at the nucleotides here, with an A goes a T, with a T then goes an A, with a G goes a C, and with a C goes a G. Now, I drew these upside down because this strand is anti-parallel. It's running the opposite direction. So this is our number 5 carbon attached to that phosphate. It's a 5' end. The OH here is attached to the 3' carbon. That's a 3' end. So the strands are opposite. The reason this is so important is because the next thing is looking at DNA replication. Then we look at transcription and so forth. And then directionality, there are going to be questions on your exam about reading DNA, making DNA, so reading it 3' to 5', making DNA 5' to 3', transcription occurring in a particular direction, then which strand is actually going to be a template strand or a coding strand. All that information comes from your understanding of this structure and the 3', 5' type thing. So you have to know this, or else the other part doesn't make much sense. Hydrogen bonds are what hold these together. 2 hydrogen bonds between the A's and T's, 3 hydrogen bonds between the G's and C's. So if you remember before when we talked about bacteria being classified, one of the things that we used to classify some of the gram-positive bacteria was a GC ratio, so how much GC they have. In this particular case, it's sort of a 50-50. But then you look at the whole genome and find out the GC content, and this is where this is coming from. So this should tie into a lot of other pieces of material. All right, so now we have the structure. We have a basic nucleotide structure, we have a single-strand structure, we have the double-stranded structure. It's typically a helical structure, though it's not ladder like this exactly, so it's twisted around, which is going to be a problem requiring the DNA gyrase or topoisomerase, an enzyme hopefully you're familiar with. So what's going to occur is the first part would be replication, right? So DNA replication starts at an origin. We call it ori-C. Typically, there's a single origin. Some bacteria can have a second origin, but usually there's one. This pulls apart the DNA in that particular region. So the DNA then gets pulled apart. The hydrogen bonds are broken. And then you get, a replication bubble, which is this whole area. And at each end of the bubble are replication forks. So there's one going, say, in this direction and one going in that direction. At each fork will then be all the proteins associated with DNA replication. So single-strand binding proteins, primase, the DNA polymerase 3 here in bacteria, all the other associated accessory proteins. We talked about now the DNA A protein being required for the process. So all these different things are happening here at a single replication fork, and you have a second set of all of them at the other replication fork working in the opposite direction. So they'd work around the cell and then come together. Again, if you had a second origin, then they'd be working in this direction and that direction, so it would shorten the time of how fast they can actually replicate the DNA so the cells can divide. So keep in mind, this is kind of a broader, big picture sort of thing. Back on your second exam, we have binary fission. So a cell divides into two new cells. The binary fission. In that process, these cells would essentially be clones of each other. And that would happen again and again. Each time before this happens, each of these cells has its own chromosome. The chromosome would have to duplicate, so there's two of them. They then have to separate. So there are other problems I talked about, that when the DNA is replicated and the whole process ends, because we have circular DNA, if you remember, in that particular lecture that I went through it, you'll have the second DNA as a loop, right? But then that loop, you can see here, is actually going to be looped, through the other loop. So the two loops are actually connected. So part of, if you think about mitosis in our cells, right, the sister chromatids are held together, they're proteins holding them together, and then they're actually pulled apart, during anaphase. So if you think of those different phases of mitosis. This is not the same, but there's this analogy between the two chromosomes made as the product of DNA replication are kind of held together. In this case, they're looped together, and they have to actually be separated from one another to then go into each cell. But then each cell would get its own chromosome, and that chromosome would be identical, if not for potentially mutation occurring. So when replication occurs, the two strands are going to be pulled apart. One strand serves as a template. Well, actually, both strands will serve as templates in this particular case. All right. They're both being, read 3' to 5', and then the new DNA, so the DNA polymerase 3 makes new DNA. That's a single-stranded DNA along a template in a 5' to 3' direction. So it'd be working, it'd be working this particular way, so not that way. So that's the kind of thing you need to know in the exam. I might ask that kind of question. There's a strand of DNA. In the direction of replication is this direction? Would this be what we call the leading strand or the lagging strand? So if I can read it easily, because the direction is the same, reading 3' to 5', moving 3' to 5', it's the leading strand. Now, you always have to read 3 to 5, or the enzyme always has to read 3 to 5. But the direction of replication for the other strand, because you remember it's anti-parallel, is going to be 5 to 3. But it can't read it 5 to 3, so that's the one that's the lagging strand. So again, I have a whole video on lagging strand. If you don't understand that, make sure you go and watch that. Find the part that I specifically go over those differences. When this happens, I said the idea is that they would be identical. But now we get into the material on the exam. So the first part is all about, gene. What's a gene? Gene codes for genetic information. Typically a functional product, which would be really RNA, technically, because some of the genes code for ribosomal RNA, some of the genes code for transfer RNA, some of the genes code for messenger RNA. The messenger RNA ones then carry the information to make a protein. But there's some kind of product is the code for a gene. The genome is the whole entire accumulation of all those genes. Bacteria have that single circular chromosome. The plasmid is extra chromosomal DNA. So not all cells have that. Some cells then have an extra circular chromosome, which would be the plasmid, and it's not part of the chromosome. It's replicated separately. So the genetic code is this, that codes for, again, RNA, and then the RNA will code for the proteins if it's messenger RNA. So you need to go through that whole process, transcription, translation. What's a nucleotide? I ask here. The base pairing I just talked about. Phosphodester bond I did. 3', 5'. Ribose versus deoxy. I basically mentioned that, saying, when you look at the sugar in DNA, these are all carbons. So the carbon. There's 5 carbons like that. Typically, they'd all have an oxygen attached to them. OH like this and an OH like this. So this one's involved in the bond that goes to the phosphate. This one's involved in the bond that goes to the nitrogenous base. This one is involved in the bond that makes the next one here. But the number 2, the 2' one in DNA, it loses it. It actually doesn't have it at all. All right, that's why it's deoxy, right? Because it's missing the OH there. But ribose, the one in RNA, has the OH there, so it's a proper sugar. This one's carbon is this one, the one that's in the ring. So know those details then. So we're sometimes coming down to the fine level of detail. Complementary strands, that's the other strand, the one that would go here with the TACG, that's complementary to ATGC. Oracy, that's the origin of replication. Here, helicase. So helicase is going to be the enzyme that pulls the 2 strands of DNA apart, breaking the hydrogen bonds. Single strand of binding proteins, the SSBP, keeps them apart. Primase makes the RNA primer. So before this enzyme can do its job, it needs a little piece of RNA to add to it. Can't build from scratch. You'll learn if you watch again the other lecture on DNA replication. It requires a little bit of RNA or a little bit of something to build off of, to attach these nucleotides to. If it doesn't have it, it can't do its job. Where am I? Helicase. Topoisomerase, or the gyrase. So that is essentially, because this is a helical structure, and as we start to pull it apart, it'll get tangled, right? Or we call supercoiling. So the gyrase or topoisomerase, what it will do is it will cut one of the strands and then unravel it around the other, relieving the supercoiling. So it prevents that from happening. Otherwise, it would get knotted, physically knotted, and then the process would be jammed up, and you wouldn't be able to replicate it. The polymerase, we said, reads and then reads along one strand, makes another strand DNA. Leading strand is the one that's reading in the same direction it's making it. Lagging strand, it can still has to follow the same rules, but it has to stop because it has to jump ahead, go behind. Proofreading should happen afterwards. So now this is what it gets into, like this other material. What could happen is, as the new strand is made, in the 3' to 5' direction this way, C, we should have the G here, right? And for G, we should have a C here. Well, for T, what if, for example, a C got put there? An error, right? That could happen, and it does happen, because this happens really fast, right? The way these are assembled and put into place, okay? So there should be proofreading that comes through and will remove the RNA, and it will remove these errors. However, if this doesn't get fixed, right, we can then have, one of these be different, say, than the other. We can have mutation. So I'll jump from here up to this part here, the mutation part. So with mutation it has different types. Okay, we can have these point mutations where a single nucleotide is changed. It should have been an A, but now it's a C. What effect will that have? We don't know until we would really see the whole sequence and how it be broken down at this moment. But it could have some effect. The types of effects it can have on here, we have a silent mutation, which is where that particular nucleotide is maybe at the end of a 3-codon sequence, and actually there's 4 or 6 different codons that all code for the same amino acid, and you may have just changed it to another one of the codons that code for the exact same thing it coded for before when that was an A, and so it actually doesn't matter. That we call a silent mutation. So there's no way to see it. It doesn't affect the protein. The protein that it's coding for is exactly the same. Everything works just the way it should. We can get a nonsense mutation, which I talked about in the lecture on those, so where this actually then forms a stop codon. So accidentally, it then codes for something that says stop. So if this were the messenger RNA, the messenger RNA that would come from reading this gene then has the code for stop way too early, and so it can't make the full protein. You can also get missense mutations where it then codes essentially for just a different amino acid. So now it's not the amino acid it's supposed to be, it's a different one, and that again may or may not have a big effect. So so that's what's going to happen. Certain things can cause that to happen. There's natural mutation. So this in DNA replication, when there's a mistake or error, that's just natural mutation. We can also have chemicals that can do this. So they can mimic what's supposed to be there. So this maybe isn't even a C, it's just something weird. It's a different chemical that is mimicking what should go there, and then later when this is a template for another strand, the enzyme doesn't know what that is. It's like, what is that one? I don't know. Is it an A, a T, a G, or C? And then it will randomly put something in place, leading to a problem, later on. You can also have deletions, right, which I mentioned in that other lecture, where there's problems that lead to an omission of a particular nucleotide, and then the whole sequence is messed up or changed from that. And that shifts then everything. If you were going to read it then in order, and you have to read it in groups of 3s, it would change that. So again, I have a whole lecture on that, talking about the frame shift mutation. Make sure you're familiar with that. As far as auxotrophs go on this here, don't worry about that. That's sort of a way of finding mutations with ones that need particular nutrients, but we'll come back to and I'll come back to this part on the horizontal gene transfer in a second. So make sure you kind of know that, then how that ties into the mutation part. As we keep moving forward, all right, so once this has happened, you've doubled the DNA, either it's exactly the same or there's a mutation, and then the cell could divide. So that's the DNA replication part. Then you have the transcription part for the gene expression. Right? So in a similar fashion, the DNA can serve as a code. Transcription factors, certain proteins will bind to the DNA, and then also an RNA polymerase will bind to the DNA. And then what it will do is it'll pull apart the DNA, kind of like that did over there, except in this case it's a little bit different. It's only going to be reading one of the strands, not both of them. In this particular case, this is the strand that contains the information, the coding strand. But that's not the one that it actually reads. It reads along the other one, the one that serves as the template. And that's because the strands are complementary. So if you want to make one with this exact sequence, A, T, G, C, you don't copy that one, because then you'll get a different sequence. You'll get T, A, C, G. If you want to get this one here, you actually have to copy the other strand, use the other one as a template. So that's what's going on here. It works along a template. The rules are the same. It reads the DNA 3 to 5. It makes the RNA 5' to 3'. Same thing, but it's a single-stranded RNA. And that gets pulled off. So what that's doing is it's doing this for a single gene at a time. Typically. Now we have exceptions to that one we talked about a little bit later when we talk about the operons and those sorts of things. But in general, that's the idea. That piece of RNA then separates from the DNA completely. The single-stranded RNA. And then the DNA would just go back together again. So the DNA just, because it's hydrogen bonds, it's just attractions, the DNA just kind of pulls back together. and then you have your RNA. So that's our transcription process, right? So you need to know the details of transcription and all the basics of transcription. And then we go on to the next part of it, which is then regulation of gene expression. So again, I'm kind of using this core part here, the DNA genes part, as the core to all the information. But then it's going to go in all these different directions, right? And we're going to add information to it. So we add in then the stuff about mutation when it's related to DNA replication. Now we're going to add in the things related to gene expression and regulation. So the transcription factors. We're going to break this down further, right? So what we have then along a single gene is that it gets broken down into regions. So we actually have the information region, the coding region. And so this is going to be, say, the RNA here that we're looking at. Sorry, that's not the RNA. It's going to be what we're going to be using to make the RNA. We're talking about doing transcription to make the RNA. So this is the coding region of the DNA of the template strand, the one that's actually going to be read. At the beginning of every gene, we have a sequence called a promoter. And that's where the transcription factors bind. Okay, if they don't bind there, then the RNA polymerase can't bind and the process won't occur. What you can have is additional regulation. Okay, so one way of regulating it is simply not making a transcription factor. So if you don't have a transcription factor, you can't read the gene. It's there, it exists, but it won't be read. So a cell has to have a signal to make a certain transcription factor to bind to a promoter to allow the RNA polymerase to copy or read a particular gene. Now, the cell itself, though, may have other plans. It may say, nope, I don't really want that to happen anyway. So there's a region here called an operator. And it's a region where a different protein can bind, a repressor protein. So the protein's called the repressor protein. The region of DNA is called the operator, just like this is a region of DNA. That's called the promoter, okay? So if that operator has a repressor protein attached to it, then it doesn't matter if the transcription factors come in, then it's stuck, so it has to be removed. And so there are different ways of regulating genes. So we get into that information, regulation of gene expression. So constitutive genes are genes that in the cell are required for basic processes like glycolysis and electron transport and metabolism and variety aspects of cell division and so on. They need to be made or the cell's going to die or it won't be able to reproduce. They have to be made. There are other ones that don't have to be made all the time, or they may not be made at all. Maybe a cell has the ability to use a certain nutrient, but it doesn't prefer to use that, so it uses glucose or other nutrients first. However, if the cell starts to starve, it will then send a signal to then read a particular gene that makes an enzyme that will then allow it to use a different nutrient. Okay, so then it will turn on a particular gene that was normally off. So then we have this terminology. Inducible genes. These are genes that are normally off. They have this repressor protein attached to them. They're not being read, but a signal would bind to the repressor and remove it, and then it turns it on. We also have some genes that are repressible, just the opposite. So repressible is where this wouldn't be here, and the cell would normally be making it like the constitutive genes, but the difference is that it can be turned off. So a signal might say, we don't need that right now, make a repressor, and the repressor will actually stop it from being read. That doesn't happen typically with the constitutive genes. It only happens with the repressible genes, so different group. Transcription factors, you really don't have to go into all the details of the protein chemistry. So zinc finger, leucine zipper, helix turn helix. Put a note in here, the lac repressor, which is one we do use as an example. It has the helix turn helix, which is the shape of that protein transcription factor. So it'd be good to know that generically, but you don't have to draw those or anything like that. Don't worry about too much detail. Another thing here is to kind of add to this, right, is that I said usually for the single gene. However, what you can have are regions of the DNA, especially in bacteria, where we get related genes. We'll just say this is, A, B, C. These are three different genes, but they all have a similar function. They all work together in a way to accomplish a task within the cell. So instead of having a separate promoter for each of them, because the bacterial genome is small, what happens is we have a single promoter for all three of them. That then creates this whole thing, this cluster of them is an operon. So the operon is a group of genes that have a related function, and they are regulated by the same transcription factor promoter. Then this process will read them all, and then later, after they're made into the RNA, you get this one piece of RNA, but then it would be cut into the separate pieces to code for each of the genes. So that would be additional processing that would occur. All right, so operons are just the clusters of them. So you just kind of need to know that. Then I go into two examples of operons, and one is for an inducible one, and one is for a repressible one. So I go for the lac operon, and I go with the tryptophan, the TRP operon. So make sure you look at those. I have a short video on it, like 6-7 minutes long. So make sure you review those in detail. So you should know the details of those. And how they all work. So I have a little bit out here about lac operon example, inducible, repressible, allolactose. That's the thing that binds here to the repressor to remove it in that lac operon example. Arginine co-repressor or maltose. So I didn't go over those if you want to study them, but I went over the TRP one. So you could substitute that in here. Regulon, I also mentioned in that lecture about being a separate regulatory region further away from, say, where the genes are, but it might affect an operon here and a different operon here and a third operon over. So not just 3 separate genes, but 3 separate operons. And not just 3, it could be more than 3 or less than 3, but numerous, several different ones. And that's called, that's different than a single operon. That's called a regulon. So it's more of a global control for the whole thing. I say cyclic AMP, that's kind of just a starvation signal. So that's one of the things that acts as a cellular signal that might trigger some of these processes. It's just, you don't have to know any details about that. The other things you don't have to know, the ribo switches we're not really getting into, the AHL, don't worry about it. Quorum sensing, just know as a term. I didn't really get to go over it, I don't think, before, but it's in the PowerPoints. There's a couple slides on it. The quorum sensing is essentially where a lot of bacterial cells, once they build up in an environment, they start to send signals out, and those signals communicate with the other cells. I did talk about this with binary fission a little bit. And essentially they can regulate the rates of cell division or rates of other types of gene expression based on signals coming from the other cells. So a whole group of cells essentially are communicating and almost making a group decision. This is the quorum sensing, as opposed to an individual cell making the decision. Okay, so the decision comes as the whole group of the cells together. And that affects gene expression. That's why it fits in here. That's what we're talking about here. All right, so that's that part. So we're coming back here though now. So that's gene expression, a little bit of the regulation of gene expression, the operons, operators, regulons, and so on. Once the RNA has been made, then you get processing. So then I have in here things like know all the basics of what comes after transcription. Okay, so the basics of, think introns and exons, 5' cap, 3' tail, the ribosomal assembly, the small subunit, the large subunit, the sites within the ribosome, the A, the P, the E site, all that kind of stuff. You do need to know what process makes the messenger RNA unique or different. So what makes the transfer RNA? What makes the ribosomal RNA? Again, I have lectures that goes into the detail on all those. You should be familiar with that. Some of the main things we added to that, or I added to that, is that for messenger RNA, for example, it has to have this thing called a 5' cap. Ours does, and most other organisms do, but bacteria replace that. So the 5' cap is a backward GTP. Its role is a binding site. So the little piece of RNA, its job is to be read, so it can code for a protein, code for an amino acid sequence. The decoder, which is essentially the ribosome, is going to attach to it and read it and then manufacture the protein. That's its job. It can't attach to it. It can't bind to it without a specific binding site. For us and for other eukaryotes, that's the thing called the 5' cap. In bacteria, that 5' cap is replaced with something else. Okay, so I call that Shine-Dalgurno sequence. So a specific sequence of nucleotides that's found here that matches up with a specific sequence, a complementary sequence that's found in the 16S ribosomal RNA. If you remember the 16S, all the way back from the first exam when we talked about classifying bacteria. How are bacteria classified? SSU, do you remember that? Ribosomal RNA. This is the same thing. So 16S and small subunit, they're the same exact thing. The small subunit of the ribosome. This is kind of where it comes in. Okay. And so they're genes that code for it, but what we're now talking about is a gene that's coding for messenger RNA that codes for a protein has to bind the small subunit, and they won't bind together without the specific sequence. So that's kind of unique for bacteria. So that's important that we emphasize that. So know the basics of the process, then also know anything that's different or unique for bacteria about the process. There aren't that many things. It's very similar from both of them, for eukaryotes and prokaryotes. Anticodons is the transfer RNA. The codons are in the messenger RNA. Wobble I kind of mentioned with the mutation bit a little bit. That's where there are multiple. So like, let's see, ACA and ACU and ACG, so on. Let's just say, for example, I don't have them all memorized. So let's just say they all code for the same amino acid. So if you change one of these, this one, to a U, it doesn't matter. They still code for the same amino acid. If you change that one to a G, it doesn't matter. That's the idea, is that with Wobble, a transfer RNA and a messenger RNA, when they pair up, even then, if a third one doesn't quite match, sometimes it doesn't matter, because the first two are really the ones that are telling you what specific amino acid it's coding for. All three are important, but you can have that wobble and still get the correct amino acid there. Ribosomal sites, amino acid structures, so make sure then the amino acids are built, the protein is built, and you get the peptide bonds between the amino acids, so make sure you kind of have the basics of that. You don't need to go into all the details of protein structure, so beyond primary. So primary is just amino acids joined together. So don't worry about secondary, tertiary, quaternary. Don't worry about that stuff, so you can remove that out. The main thing you need to know is that the protein, the polypeptide that's assembled, has to be folded. So after it's made, it has to be folded into the right shape, and that is controlled often by molecular chaperone proteins, so other folding proteins. And that's another level of regulation. So you can essentially say, I'm going to read this DNA because I have the right transcription factors that bind to the DNA, and then I'm going to read this little piece of a particular gene. The RNA polymerase is going to read it. It's going to make this single-stranded RNA. But you can also stop that. You can control it with repressors. So the big part of the exam too is this regulation of expression. As I go through the lectures, they're kind of in that order. Regulation of expression then involves all these different levels. So they go from regulating it at the DNA level, that it will not be read and turned and copied to make a piece of RNA. That's the transcriptional regulation. But if you do transcribe it, you can still have regulation. So you might not modify the RNA properly so that it won't be used. You might bind to it and shut it down, or not bind to it. If it is used, okay, and then we read that information and then make the polypeptide, well, the ribosomes have to bind to it in a certain way. They can be potentially blocked. But if you make the polypeptide, it still has to be folded. So you can have translation occur, going from RNA to the protein, but still not have it be expressed, because when it's expressed, the protein is made and carries out its function. So when we talk about genes and gene expression, we're talking about taking information from the DNA, creating a piece of RNA from it, using that piece of RNA to make a particular protein, and then having that protein carry out a function. If it doesn't carry out the function, then the gene technically isn't expressed. It could be a structural function, doesn't have to be an active thing, but depending on what the protein is, it may not then have that function unless it's folded properly, so it doesn't receive the right shape. So control occurs at many different levels. I know there's a PowerPoint slide that gives you an overview of all those as well. I didn't talk about the horizontal gene transfer. So coming back into this, so cells in general, aside from mutation, might be just clones of one another. So cells divide, cells divide, cells divide, gene replication. The process should be accurate every time, and there shouldn't be much error. If there is, then it can lead to things that are harmful to cells, or maybe things that are beneficial, just kind of randomly. So how do cells become unique? How do cells become different? How do we get many different species of bacteria if there's no genetic exchange? So to get genetic exchange, we have the HGT represented on here, the horizontal gene transfer. So that's where the one cell has genetic information that's unique, and it can then pass that genetic information to a new cell. Pass information, and that's genetic information. And so one of the main bacterial mechanisms for doing this is called conjugation. So that's the one to focus on. Bacteria have a plasmid separate from the chromosome, so extra chromosomal DNA, that codes for the production of a pilus. So the pilus then is a structure that will then connect the two cells, and it doesn't pass the whole plasmid. The plasmid is double-stranded DNA as well. It'll pull it apart, but it'll take a single strand of the plasmid and pass that single strand into the other cell. And then in the other cell, DNA replication will occur of the other strand, the complementary strand, and you'll get that plasmid. So we have F plus cells are the ones that have that plasmid. F minus cells don't have it. An F plus cell, if it passes the plasmid to the F minus cell, it will become an F plus cell. Where it gets tricky is there's another version of this, the HFR. So if you go back in the lecture, I talk about these in the conjugation lecture. Some cells are HFR, so that means high frequency of recombination. And what happens there is that the bacteria have this F plasmid gene, but it's not in a separate plasmid. Well, they have the gene to make the pilus, right, to pass on information, but it's actually part of the bacterial chromosome. And so I'm going to erase a little bit of this just so I can give you this example as part of the review, okay? So, because this one is tricky, I know a lot of people end up getting very confused on this one and what happens. And so, again, you have my video where you can stop it or rewind it if you don't understand what I'm saying, but maybe this will give me a chance to restate what I said before. And maybe it will make more sense this time if it didn't make sense to you before. So I'm going to redraw that. Okay, so here we have this is a cell. Here is the cell's chromosome. This is its main bacterial DNA. It's genetic information. And then we have these genes that code for the pilus formation. So now those genes can be expressed, and what will happen is it will form a pilus. Over here we have a cell, a recipient cell. Now, if we had to label these cells, this one is called the HFR cell. This one is still going to be the F minus cell. What will happen is that, again, this is double-stranded DNA, so it's going to snip some of this and then feed through the pilus DNA. The thing is, it's going to feed through it part of just the bacterial chromosome, and it may get to the section with the genes that code for the production of a pilus, but usually it doesn't. So usually when this happens, this cell over here, who has this genetic information, gets passed on new genetic information. I've got a different color here. Easier if I put that down. So that's going to come along here. This all connects, comes into the cell, and then we get this piece of information. These are bacterial genes from the chromosome of this other bacterial cell. What will happen is that they will then usually have DNA replication occur along it to make it double-stranded. Then that double-stranded piece lines up just kind of like in meiosis. So if you think about meiosis and the process that we have in our cells that make our gametes, the process called crossing over. Okay, that's recombination. So high frequency of recombination. Recombination means then genes might be swapped. Okay, so what will happen is some genes, so maybe even not all, so it could be the whole thing gets inserted in there, or it could be pieces of it, but basically some of that information from the other bacterial cell gets put into this chromosome. It's not the F plasmid, so I probably shouldn't have put the pink there, because that was when I was representing. I'll make it a different color. There you go. It's not the F plasmid. So this cell remains F minus. It stays F minus. It doesn't become HFR because it didn't receive this full set of genes to make an F plasmid to pass on to other cells. That's typically what's going to happen there. So, but it does receive new genes from the other bacteria. Right, so there's a lot of recombination and new genetic information being swapped between those cells. So that's why it's kind of interesting and important. Right? So make sure you know that's the part. Those are the parts with lots of detail. And then the other thing with a lot of detail is transduction. So it was specialized and generalized transduction. That's where a virus, a viral infection, it's called a bacteriophage. So that's where, there's some of this. It's going to be, I think, the last thing in the review, and I'll look at the questions on the bottom. But again, now your exam is going to be all primarily multiple choice. There'll be a couple questions for you to submit that are written out that you'll submit separate from the main exam. Okay, and those will be more of these essay kind of questions, so you'll have more time to do those. But you have a time frame in order to submit them, and they'll be a little more complicated than they'd be on a normal exam because you're obviously it'll be open book. You can look stuff up. So a virus, which was now going to be the topic of the final exam. A typical virus that affects bacteria is called a bacteriophage. P-H-A-G-E, okay? And so what will happen is that bacteriophage attaches to a bacterial cell. The way a bacteriophage works is that it has in its capsid, which is a little protein shell, genetic information, either DNA or RNA. And it then injects that into the host cell, the bacterial cell. The bacterial cell has its own genome. But when that occurs, along with it comes proteins and codes for proteins that in many cases will destroy the bacterial chromosome, and it will begin the replication. So you'll get actually replication of the viral genome, and you'll get gene expression. So the genes being copied from DNA into RNA, and then proteins made, which would be the viral proteins, which would then make copies of the virus, and then little by little it'll start making many more little viral particles inside the cell. And then those viral particles get the replicated DNA into them, and then eventually the cell bursts, and then they spread. Okay, and they cause damage. They kill that cell, and they spread to other cells, and so on. Now, when that happens, so okay, that's the whole process we're going to talk about in the next exam. So you're going to have to learn. This is material that's on exam 4 that you're kind of learning now. It overlaps. It's a big overlap between the two of them. The part that you want to be focused on for this current exam is not necessarily the viral life cycle phase. It's so how does a bacterial cell get new genetic information from this? So far we just see it, virus affects the cell and the cell dies. So that's not part of horizontal gene transfer. Where's the horizontal gene transfer come in? How that comes in is that there are some types of virus that their DNA is actually at first inserted into the host cell's genome to start off with. And then after a period of time, the virus is triggered to replicate. What happens then is it gets cut back out of the genome. But when it gets cut out and the rest of the host cell's DNA is destroyed, usually what happens is some of the host genes, I'll just erase the whole thing and redraw it so I can make it clear, some of the host cell's genes stay attached to it. So those are the bacterial genes. This is the whole viral genome. But now we have a new piece. So that means that when the replication occurs and we get all these bits. We actually have bacterial DNA in all these. Again, for that cell, it didn't do anything with horizontal gene transfer. But now, as those viral particles burst out, and they go on to now infect other cells, what we have inside this cell here is the viral genome that it makes it a virus and does what it does, and we have bacterial genes. And so we get this new bacteria over here. Here's its genome, its chromosome. When this information is injected into the cell, if it then gets incorporated like this, so spliced in. There. Now this particular bacterial cell, until those viral genes are expressed, this bacteria now contains genes from a different bacteria. That's the horizontal gene transfer. And it can then express those genes, and they may have a function and they may not have a function. They may be a redundant function, or they may be something new. But that's the way this particular bacteria can get that information. And then the other thing you can think is that, well, but the bacteria is going to be killed anyway, so what? You know, it's like, how does that contribute to a broad aspect of the concept of bacterial genetic diversity? And this is because something we didn't really get into at all, and we'll talk about a little bit later, is bacterial cells have their own sort of immune system. So you have probably heard of CRISPR-Cas9, which are proteins that humans use in editing genomes of different organisms, including humans, and hopefully trying to correct certain genetic disorders using this, and a variety of other sorts of things that could be used in medicine. So it can be it's a specific protein that can cut DNA, remove pieces of DNA, but also be used along with others to then replace those with other pieces. We didn't develop this. This came from bacteria. This is part of a bacterial, essentially, immune system where the bacteria can say, hey, I'm reading this information, and this information here is not bacterial genes. Those are foreign, and so I'm going to cut them out and splice them away. And so it can actually get rid of them and destroy them and then reheal itself. So the cell's not going to die from a viral infection. But because it was infected at one point and those genes went through this process of inserting themselves into the chromosome, the genes from the other bacteria would remain, and now it's changed and it's not going to die. So we get this idea of the horizontal gene transfer. The bacteria picks up new genes from another bacteria, but the genes are transmitted through a virus. Okay. And this is the specialized transduction. Where they are very specific genes, and then every single viral particle would have the exact same ones. Generalized is where the chopped up DNA has little bits and pieces in it, and they all have little different pieces of bacterial DNA. So it's not the same, and they don't typically insert themselves into the genome. So it's a different way that those cells then may take up the piece of bacterial DNA and have it be inserted into their chromosomes the way they would from the other process, the process of transformation, where you get DNA from the environment. So it may not do that. I think that's everything. So you should know all those terms, basically, except for the ones I said don't worry about. So, and then as you go into these questions, I'll try to go very fast through them. Process of DNA replication, I already talked about. Nucleotides, draw them, or be able to describe them at the very least, or answer questions about their structure in a multiple choice type fashion. Identify the components of the replication forks. That's what I was talking about. Helicase, primase, single strand binding, proteins, the topoisomerase. That's what that means. That's the enzymes involved in DNA replication. That's pretty much the same thing. translation. So we're talking there about protein synthesis. So that's where the ribosome, small subunit, large subunit, come in. The transfer RNA comes in. The messenger RNA is being read. So make sure you know that. Amino acid and peptide bond. I'm probably not going to have super sketches in this, so don't worry about that too much. So be familiar with it, but don't go overboard on drawing those. What's structure transfer RNA? You should know that. Know what it does. What are ribosomes? You should know that. Know what it does. Bacteria has the 16 and 23S ribosomal RNA for large and small subunits. Know what the ribosomes are, the parts of the ribosomes. Make sure you know that. Translation. So again, it's all translation questions. They're all pretty much an overlap. And make sure you know, I'll add in here, anything that's unique or different about bacterial replication, transcription, or translation, like the Shine-Dalgarno sequence, for example, something unique, or something unique about the origin of replication and DNA A protein binding to the ORI-C specifically. That's something unique and specific for bacteria. Make sure all those specific things you really hit on first, and then you know the big picture of all these processes kind of second. Genetics of bacteria and archaea, so we didn't really go over archaea much here. Most of it's the same really for bacteria and archaea. One thing I'd say that's different to point out, no introns here. So when we went through the process of DNA being read, transcription into RNA, in bacterial cells there are no introns to remove, but archaea do have introns the way our cells do. So there's a difference between them. So when you learn about post-transcriptional processing, so the removal of introns and so on, that still holds true for archaea, those types of prokaryotic cells. So it's not only an exclusive human or eukaryotic thing, it's also for the archaea. So that's why you need to know those things as well. Mechanism of genetic exchange, that's what I just talked about. Horizontal gene transfer, the F+, F-, and the HFR cells. Make sure you know all that. General specialized transduction, and all that type of stuff. Here I do actually mention what are CRISPR and its role in prokaryotic cells. So I basically just talked about that right there really quickly. So you don't have to know much detail about this. Some of you have done your literature projects and include CRISPR in it, so it's good if you know more about it, but I'm not going to ask anything much about it at all, except maybe one reference to it. The gene regulation part, so this is the then gets into more detail. Modes of metabolic regulation and gene expression. I'm not even sure what I'm asking with that particular question. Really more of the big picture type of thing, of the levels of where regulation occurs. Like I already mentioned that. Transcriptional level, translational level, post-processing level. So there's all those kinds of things. DNA binding proteins, talked about those. That's what's going to bind here to the transcription factors and these other regulators that can bind to the operator or the promoter. Those are all DNA. Remember, those are DNA segments. Negative positive regulation essentially means like turning on, turning off. I wouldn't worry about those terms as much as the inducible, repressible. So look at those. So inducible means it's usually off and you can turn it on. Repressible means it's usually on and you would turn it off. And that could be for a single gene or for the operons. So make sure you know what the operons are as well. And so that's all there. Example of lac operon example. Make sure you know it. Don't worry about the additional stuff, the specific genes and cap binding site and all. So not the details I gave in my presentation on is all you need to know. Catabolite repression, don't worry about that. So that was number 10 here on gene regulation. Don't worry about that question. Explain how catabolite repression depends on activator protein. Again, don't worry about that. That has to do with starvation and so forth. It's related to the lac operon, but right now if you know the basics of it, that'll be good. Positive and negatively controlled, don't worry about that. We're just a specific example because it gets more complicated. I just gave you the very introductory version of it. There's a lot more actually to it and it gets very complex, but just know the basics. Two-component regulator system, don't worry about that. So number 13. And number 14, I just mentioned the quorum sensing, so it's just like as a term. That's all I mean. There's no big process or anything that you need to know or be aware of, no details or anything like that, just a term. That should conclude everything that's on this exam. Again, the good thing for you is that the fourth exam, the final, has to do with virology and the immune system. So it's a very relevant, timely thing, fortunately, at the moment in time. So it should be very interesting, hopefully, for you to study that. And the second part is there's a lot of overlap with this material and the whole viral part of it. So there's the general specialized transduction. That's kind of goes forward. We go into different perspective from the virus, the structure of the virus, and all that kind of thing. So, but there's a lot of double-stranded DNA, single-stranded DNA, single-stranded RNA, double-stranded RNA. So this concept, the structural stuff, becomes still important in all the viral stuff toward the end. So you have to know it now because you're going to have to know it then for the last material. So just make sure you do study this. Study for the exam as if it's just a regular exam. You're taking it in a classroom with a specific time with an instructor there. You're not really supposed to be looking at your notes. You're supposed to be taking the test. That's how you should study. The exam is 45 multiple choice questions. I made it right now. And you're going to have a time limit. It's 50 minutes. So you have a little more than a minute per question. It shouldn't even take you 30 seconds per question. You basically have to read the question, and then you read the answers. When you find the right answer, you just click it and you just move on. So you should have plenty of time to read and do it. You probably would have enough time to do it and then hold off on submitting and then double check something that you were not 100% certain of. So that's possible. So your bonus essentially in this is the ability, potentially, if you study ahead of time and are very prepared, that you could go back and actually look up probably a few things before your time runs out. It will auto-submit. 50 minutes, it submits it for you. If you only answer 20 questions, you'll get those 20 scored and zeros for the remainder. So you need to be ready as if you're taking an actual test. And so, but like I said, there will not be enough time for you to not study any of this and then just say, well, it's basically open book test. I don't really have to study. I'm just going to come and sit down on my computer. I'm going to start up the test. I'm going to have PowerPoints and things there, and I'm going to Google stuff, and I can answer the questions. You will get several of them answered and probably answered correctly, but you probably will not be able to finish. I doubt you would be that fast. If you had a team of people helping you, probably can go through it, but you won't do it alone if you don't study. If you do study, most people finish these types of exams. I've given them in the past. It's a 50-minute period. Most people finish like 20 minutes because I can see exactly how long it was before people submitted, and a lot of people submit far before the end of time. So I know it's plenty of time for the majority of people. If you're someone who needs more time because you have that specifically outlined or designated from some other aspect earlier in the semester, I'll have that worked out separately. But overall, it's a 50-minute period. There's a separate part for 10 more points. That's a written part, and so that'll be separate. It'll be like exam 3 part 2, and it'll be something that you do and then submit like you've been submitting the labs. You'll submit it like that. Hopefully this helped. I know it was much longer than normal, but I just wanted to make sure you had some kind of actual review for this exam. So we're treating it as normal as possible, like a normal material, normal exam, but now you're just going to be doing it online. So you'll just go into the Blackboard, you'll find the exam, and you can do it at any time of day, over any of the days it's available. It's available for a 10-day period. So technical problems or other things as far as scheduling shouldn't affect when you do it, but I would encourage you to do it as early as you can in that time period. Do not wait to the last day, because that's time that you could be starting in the last material, or working on the labs or something else. So that'll be it.